All right. I think it should be working now. The YouTube one.
Yeah, the YouTube is working.
One chat, I can't share my screen. Is there a way to enable that?
Let me see.
Javier, are you the host?
Yes, I'm.
Do you know how to make him host?
Yeah, can you try now?
All right, awesome, got it.
Okay. Yes. I think you should be able to share your screen now.
All right, do you want me to put it up?
Yes.
All right, cool.
It works, yeah.
Okay, perfect.
Can you see the slides all right?
Yes.
All right, cool.
Okay. I think we're fine. Okay. All right, so I think we can start now. Sorry about the technical problems that we had at the beginning. Okay, so for some reason, I don't know where this comes from, but I'm hearing myself. So I don't know if.
What was that, Javier? What do you mean?
You're on mute, Javier.
You're on mute, Javier. You're on mute.
You're on mute. Yeah, you should be able to sign in.
Oh, should I sign in? Because this is
Javier, you're still muted if you're
You're
talking
mute.
to us.
Yeah, you can. We cannot hear you.
No?
Yeah.
Now it's good.
Okay, I was trying to figure out where this kind of back sound was coming from. All right. So welcome everyone now for real to the inaugural seminar of this INCEPT series. So these seminars will feature fellow neurobiology researchers not affiliated to Harvard, who are currently either in the very late stages of their postdoctoral research or, as in we have today, have very recently become independent. So overall, the goal of this program is to promote scientific exchange and networking between young scientists. We aim to provide a bit of a platform in which speakers can gain more scientific visibility, meet established faculty, postdocs, and grad students within HMS, and vice versa. So we believe that also this platform will be an excellent chance for postdocs and grad students here to learn from a successful young scientist. We believe that some of the speakers that we will feature will actually become leaders in their respective fields of research. And also to gain insight and perspective into other challenges and opportunities to become a PI nowadays. And actually to dig deeper into this topic of how to become a PI, we are hosting an open discussion at 4 p.m. It's the same Zoom link. And during this session, we will, of course, also be addressing any scientific question that cannot be addressed during the talk right now. I encourage all of you to join us. And even if this discussion was initially advertised for postdoc and grad students, any faculty that wants to join to discuss science or discuss like this transition to PI is of course very welcome. So INCEPT is an initiative of the Neuropostdoc Lab, and it's organized by Yasmin and Giacomo from the DMEC Lab, Charlene from the Orifice Lab, and myself from the Kaiser Lab. And of course, we had a lot of help from the department, so Janine, Soha, Jonathan, thank you for this. This first INCEPT series will only have 3 seminars. They will all, of course, be virtual. So the one today, one in February, and one in March that we will later on advertise. And they all feature speakers from Massachusetts institutions. And our goal eventually is to expand to the whole country in future editions. So, and while we will try to acquire the means to actually bring the speakers physically here to HMS for better interaction once, of course, COVID is over. And of course, to get there, we will need the support of the community, and the best way to support this initiative will be, of course, by attending and participating in these meetings and discussions. We're also streaming by YouTube, so those who are following by streaming, if you have any questions, just type a comment there, and we'll try to get to. So now I would like to introduce you to the speaker of the day, Joel Blanchard. Joel did his grad research at the Scripps Research Institute with Professor Christine Baldwin, working on stem cell research in the context of neuronal reprogramming. After that, he moved to the lab of Li-Hu Tsai at MIT, where he kept his interest in stem cell research and reprogramming. In this case, applied to the blood-brain barrier and Alzheimer's disease. And just to mention that he currently has a position as an independent researcher at Mount Sinai in New York. So with this, I will just let the speaker start his presentation.
Thank you, Javier, for the kind introduction. I'm really excited to be here today, and although virtually, I share some of the data that we recently published and an unpublished story, and also then to meet with a bunch of people and discuss some of the challenges of doing research in these times. So I'll tell you 2 stories today. One that's published, as I mentioned, about the effects of an AD risk factor in the blood-brain barrier. Then I'll tell you another unpublished story about how the same risk factor affects another tissue, myelinated axons, and how that might also predispose people to Alzheimer's disease. I'll start with just some background to give you a broader context of the type of research that I do. And that's the slide here. So most of us will live cognitively normal lives into our 80s and 90s, but unfortunately about 1 in 10 of us will experience cognitive impairments starting in around 65 to 70 years of age. And women are about 2 times more likely to have cognitive impairments than men are. And currently this is quite a big problem. If you look at the most prevalent form of dementia or Alzheimer's disease, it's estimated about 5 million Americans currently have Alzheimer's disease. And this is really a growing problem. By the year 2050, the number of people with Alzheimer's disease is estimated to triple to about 16 million people. When we talk about Alzheimer's disease, we're really talking about 2 separate diseases. There's an early onset Alzheimer's disease that affects people starting in their 40s and 50s. And for early onset Alzheimer's disease, the genetics is really well understood. There are mutations in the genes that process or encode amyloid protein, and this is really what drives early onset Alzheimer's disease. But for the vast majority, nearly 95% of Alzheimer's disease, the age of onset's about age 65, and we really don't understand the genetic underpinnings, and currently we have really no effective therapies. We do have some clues as to what is driving the Alzheimer's disease through genome-wide association studies or GWAS studies. For instance, if you look at this loci here, ApoE, it's one of the strongest risk factors or genetic correlates of Alzheimer's disease. And I'll be talking predominantly about this risk factor because it's found in and approximately 60% of late onset Alzheimer's disease cases. And so to dive into it a little bit more, the humans have 3 common variants of the ApoE gene that you can inherit from your mother or your father. And the most common form is called ApoE3, and this has a neutral effect on Alzheimer's disease. But a single nucleotide polymorphism or a single amino acid change creates a variant called ApoE4 that I'll talk more about. And if you have 1 copy of this, your risk for having Alzheimer's disease is 3 to 5 times greater. If you have 2 copies of this, you have nearly 10 to 12-fold increase incident risk of Alzheimer's disease. And I won't talk about ApoE2, but this is also a single nucleotide change from ApoE3, and this has a protective effect against Alzheimer's disease. So I think this bidirectional risk and protection effect really illustrates the importance of this loci or gene in Alzheimer's disease. What's really striking about ApoE4 is it's a risk factor. You can be homozygous or have 2 copies of ApoE4, but you can live a cognitively normal life into your 80s and 90s and never experience Alzheimer's disease. And so this really suggests that the genetics of Alzheimer's disease might be polygenic, that ApoE4 needs to act in conjunction with other risk factors to have a pathogenic effect, or that there are unknown resilience factors that protect against Alzheimer's disease if you have a ApoE4. But this not clearly understanding the genetics that really drives Alzheimer's disease has led us to, and the long nearly decades that it takes to develop Alzheimer's disease or the course of the pathology, has really caused our therapeutic approaches to focus on the late stage symptoms. So when people start to present with cognitive impairments, we start to provide therapeutics. And right now, the best therapeutics we have are really targeting the major symptoms of the disease, the amyloid buildup. But through understanding how the genetics contribute to this increased risk and start to drive the disease pathogenesis, we might be able to identify earlier interventions that are genetically informed. So a lot of my focus of my research is trying to understand what these genetic factors do, or what lifestyle differences such as dietary impacts do to contribute to Alzheimer's disease. And over the past few years, we've got some insight into this by looking into the brains of Alzheimer's disease patients and comparing them to normal patients using single-cell transcriptomics profiling. And this has really been able, this is a paper published by my postdoc mentor, Liwei Tsai, in 2019, and we've been able to identify single gene expression changes in different cell types that correlate with Alzheimer's disease. But these gene expression changes are really at the end of life. And so we know how each cell type in the postmortem brain ends up, or at the end of Alzheimer's disease. But taking these gene association studies and the end of life gene expression profiles at single cell resolution, it's a long ways from understanding how we can use these combine these 2 pieces of information into therapeutic approaches, because we don't know if these are just things that correlate with the onset of Alzheimer's disease, and we don't know the order of events. Is this the last thing that happens, or does it really drive the disease pathogenesis? And one of the analogies I like to use to explain this situation is it's similar to a plane crash where investigators observe that the wing is broken off the plane, but they don't know if the wing broke off because the plane crashed, or the plane crashed because the wing broke off, or because the wing broke off. And what's really helped investigators understand plane crashes is the black box, or the on-flight recorder. And so really, to summarize my postdoc studies, it's really been trying to build an on-flight recorder by combining stem cell biology, neuroscience, and tissue engineering with single-cell transcriptomics to understand Alzheimer's disease pathogenesis by playing and replaying the pathogenesis in vitro using tissue-based models. And to illustrate this more, I'll tell you two different stories about the blood-brain barrier and the myelination of how we're building models of the human brain in a dish to dissect the mechanisms of Alzheimer's disease. So I'll start with the blood-brain barrier story and give a brief background on that. The human brain is a highly vascularized organ. It's estimated if you stretch out all the capillaries in the human brain, they'd span approximately 400 miles. And every neuron is in an intimate interaction with the brain vasculature. And unlike the peripheral vasculature, the cerebral vasculature forms this specialized barrier that really not only delivers oxygen and nutrients to the brain, but protects it from peripheral insults. And so it's really emerged over the past few years that Alzheimer's disease might emerge because the blood-brain barrier breaks down and the neurons and stuff aren't getting enough oxygen and nutrients, and pathogens are infiltrating the brain. And so the blood-brain barrier is really composed of three separate cell types: the endothelial cells, pericytes, and astrocytes, which extend specialized and feed onto the capillaries. And so we want to know if we could recapitulate this blood-brain barrier in vitro so that we could understand how AD genetics influences its susceptibility to pathology. And so we started by taking human iPS cells and optimizing differentiation protocols for each of the cell types: brain endothelial cells, pericytes, and astrocytes. And on the right here is just representative images for each of the three cell types. We went a little bit further. We wanted to make sure that at a transcriptomics level, that what we were making in a dish actually reflected what was in the human brain. So we used single cell transcriptomics to isolate each of the cell types from the human brain. And then this allowed us to compare the whole transcriptome of our iPS-derived, for example, pericytes to in vivo human brain pericytes. And what we observed is that the iPS-derived pericytes had a high degree of similarity to the in vivo human brain pericytes, and were significantly different from other cell types such as smooth muscle cells, astrocytes, and microglia. And so this suggests that the cells that we were making in a dish might have some predictive fidelity about what's going on in the human brain. So we're interested in how a tissue is affected during Alzheimer's disease pathogenesis. So we tried to make a tissue-based model by encapsulating all three of these cell types into a hydrogel matrix, which allows them to interact and self-assemble. And after a period of about 4 weeks, what we observed is that the endothelial cells, stained here with the cadherin shown in green, actually formed tube networks that were reminiscent of microvessels in the brain. And what was even more interesting is when we initially put the pericytes into the culture, they were homogeneously dispersed, but the endothelial cells were known to secrete a growth factor, PDGFBB. And so even after about 2 weeks, we saw the pericytes or mural cells to start to line the capillaries in this in vitro culture system. And we've started similar things with astrocytes, just trying to verify that the model system has anatomical properties of the blood-brain barrier in vivo. And so astrocytes localize end feed, as I mentioned, and on those end feed are specialized transporters for regulating water volume in the brain or other solutes. And what we observed in the in vitro blood-brain barrier, or IBBB, is that aquaporin 4 was expressed in our model system and largely localized on astrocytic end feed along the in vitro vasculature. And so we've done a lot of studies like these, and I won't go through them all because the work's published, but essentially we think we can mimic certain anatomical, molecular, and physiological properties of the blood-brain barrier in vitro. And we're really interested in applying this model to disease. And so we're interested in Alzheimer's disease, and one of the primary cerebral vascular pathologies in Alzheimer's disease is this pathology called cerebral amyloid angiopathy, or CAA. And what that is amyloid building up along the capillaries and vasculature in the human brain, shown here in blue. And it's estimated that about 95% of Alzheimer's disease patients have CAA pathology. And so we wanted to know if we could model CAA using our our in vitro system. And we did a bunch of studies to show that the cerebral vasculature doesn't produce a lot of amyloid, and it's really known that the amyloid in the brain comes from neurons. So what we did is a conditioned media experiment where we took NPCs, allowed them to differentiate into neuron and glia, and then collected the media and put it on our IBB. And using patient-derived IPS or NPCs, we could take NPCs from a healthy person that has low amyloid in the conditioned media. And when we did that, we observed that there wasn't a lot of amyloid immunoreactivity along the vasculature, shown in green here. When we took conditioned media from NPCs from a familial AD patient that has high amyloid, what we observed is that there was a lot of amyloid immunoreactivity or buildup along the vasculature, shown here in green. And so this suggests that we can model aspects of this pathology using our in vitro system. So the next thing we really want to ask is whether we can model the genetic susceptibility for CAA in a dish. And as I mentioned, ApoE4 is a risk factor for AD, but it's also a risk factor for CAA. And so to ask this question, we use CRISPR editing to generate isogenic sets of iPS cells that either they're homozygous for E3 or nearly identical iPS cells that are homozygous for E4. We differentiated them into the three vascular cell types and then encapsulated them into the hydrogel matrix, allowed it to mature for a couple of weeks, and then exposed it again to a high amyloid environment and asked if there were differences between the E3 and E4. And really encouragingly, what we saw is when we took these genetically identical IBBs, the E3 again had some amyloid building up along the vasculature, but the ApoE4 IBB just had a ton more amyloid building up along the vasculature. And so to make a long story short, is we did this experiment multiple different ways. Here I'm showing using two different CRISPR editing strategies. We can edit the non-risk E3 into an E4, or we can do the reverse, edit another iPS cell line that has ApoE4 into an E3. And regardless of the editing strategy, consistently we saw if you have two copies of ApoE4, this leads to a high amyloid state. And we've done this with non-edited lines that are heterozygous and found that even one copy of ApoE4 is sufficient to significantly increase the vascular amyloid accumulation, which is reflective of what's seen in the clinic. And so we're able to model this cerebral vascular pathology in a dish. We wanted to know if we can use this model to start to dissect what are the key cellular and molecular events that are driving this. And so the first question that came to mind was, is ApoE4 required to be expressed in all of the cell types to drive this pathology, or is it only some of them? And so what we were able to do with this engineered model system is what we call an isogenic mix and match experiment. I've showed you we can express ApoE3 in all of the cell types, or ApoE4 in all the cell types, but we can do everything in between. We can have each combination of ApoE3, ApoE4, either pairwise or singular. And so there's 8 permutations. We made all 8 permutations, and that allowed them to mature and then exposed them to high levels of amyloid again. And what we observed is that, again, all ApoE3 had low amyloid buildup. The all ApoE4 had high amyloid buildup. But then the different combinations had different levels of amyloid. So we quantified all the amyloid and basically asked which combinations are similar to the all E3 state and which combinations are similar to the all E4 state. And to summarize this more graphically, essentially what we found is that if pericytes express ApoE4, this consistently led to a low amyloid state. Express ApoE3, this consistently led to a low amyloid state. And if pericytes express ApoE4, this consistently led to a high amyloid state. And so this and a handful of other experiments that we did really suggest that the expression of ApoE4 in pericytes, it plays a causal role in the increased cerebral vascular amyloid accumulation. And so we were able to pinpoint the pericyte, but then we wanted to dig into the molecular mechanisms underlying this more. And first, we wanted to know whether it's the physical interaction of the parasite with the vasculature, or is it something that is secreted by the parasite. So for this experiment, we did basically another conditioned media experiment, only this time using parasite conditioned media, reasoning that if it's a soluble factor, that the ApoE4 parasites would be able to drive up cerebral vascular amyloid accumulation in the ApoE3-IBB. That's what we found, is that if we expose the ApoE3-IBB to E3 parasite conditioned media, this doesn't lead to an increased amyloid accumulation. But if we expose the E3-IBB to a ApoE4 parasite conditioned media, this leads to increased amyloid accumulation. So this suggests that ApoE4 parasites are secreting a soluble factor. And so to really get an idea of what that soluble factor is, we did RNA sequencing to compare gene expression profiles of isogenic ApoE3 parasites and ApoE4 parasites. And we were shocked to see that there was dramatic gene expression differences between ApoE3 and ApoE4. It's only one amino acid change, and there are nearly 2,000 genes upregulated in ApoE4 parasites and 2,000 genes downregulated in ApoE4 parasites. So there's a lot of data to sift through here. But what really struck out to us is that in ApoE4 parasites, the expression of ApoE itself was upregulated nearly twofold. And this was particularly interesting because ApoE is known to be a secreted protein, so it's a soluble factor. Plus, it's known to interact with amyloid. So we hypothesized that this increase in ApoE expression might be driving the pathology itself. And so we wanted to make sure that this wasn't just an artifact of our iPS-derived parasites and some in vitro observation that we're making. So we went to human brain slices from the hippocampus. And what we observed when we stained for the vasculature and parasites along the vasculature is brain hippocampus from ApoE4 carriers had dramatically increased ApoE expression relative to non-ApoE4 carriers in parasites. So this suggests that in the human brain, ApoE4 parasites have increased ApoE expression. And so we've done a number, I won't go into all of this data here, but we've done a number of experiments to demonstrate that increased ApoE expression has a causal role in increased amyloid accumulation. So we've used a CRISPR knockout strategy and found if you knock out ApoE, this reduces vascular amyloid accumulation. If you immunodeplete ApoE using antibodies, this reduces cerebral vascular amyloid accumulation. And if you do the reverse, you add in recombinant ApoE, this dramatically increases the cerebral vascular ApoE accumulation. So this really suggests it's the levels of ApoE that are driving the vascular amyloid accumulation. And so we know that ApoE4 parasites are secreting more ApoE. And so if we can determine how they're secreting or why they're secreting more ApoE and suppress this pathway, we might be able to reduce the vascular amyloid accumulation. And so the next question that we focused on is, why is ApoE upregulated in ApoE4 parasites? And so we went back to this transcriptome or RNA sequencing data set, and we reasoned it was really a transcription factor mediated event that's leading to upregulation of ApoE in ApoE4 parasites. So we isolated all the transcription factors. There are nearly 100 downregulated transcription factors by ApoE4 and 100 upregulated transcription factors in ApoE4 parasites. We asked if any of these transcription factors had been reported or known to bind ApoE regulatory elements. And in fact, two classes of transcription factors really stuck out. The CEBPs and NFAT pathways were both differentially upregulated in ApoE4 parasites. So just to make sure this wasn't an in vitro artifact, we also looked in the human brain and post-mortem hippocampal parasites. And what we found there, again, is that in parasites from ApoE4 carriers, NFAT-C1 and NFAT-C2 were both upregulated compared to non-carriers. So this demonstrates that in ApoE4 carriers, there's upregulated NFAT, which we hypothesized could be driving increased ApoE expression. And so to test this, we had to learn a little bit about the NFAT pathway. And so NFAT in an inactive state resides in the cytosol because it's phosphorylated. When this pathway becomes activated, a phosphatase called calcineurin actually cleaves off the phosphate, which allows NFAT to translocate to the nucleus, where we are hypothesizing it interacts with regulatory elements of ApoE, allowing its upregulation. And so there are well-established inhibitors of calcineurin, cyclosporin A and FK506, that can block the removal of the phosphate and the nuclear translocation of ApoE. And so we treated the pericytes with this, and then with cyclosporin A and FK506, and encouragingly what we found is that when we treat ApoE4 pericytes with cyclosporin or FK506, not shown here, this dramatically reduces the ApoE expression. And the animations were a bit messed up, but we also treated the in vitro cultures with this, reasoning if ApoE expression is downregulated, this will lead to a removal of amyloid. And so in the control IBB, we see a lot of this green staining, which is the amyloid buildup. But when we treat the IBB with cyclosporin FK506, then apply the amyloid, less amyloid builds up on the ApoE4 IBB. So suggesting that inhibiting this pathway can block cerebral vascular amyloid accumulation. We're really excited that we could do this in a culture dish, but the real goal is to translate this to a living organism and into humans. And so we immediately moved to a mouse model where the mouse ApoE has been removed and replaced with human ApoE, and the mice are crossed to an AD model called the 5XFAD mouse. And at 6 months of age, these mice develop CAA pathology. And so we treated these mice either with a vehicle control or 10 mg per kg of FK506 or cyclosporin for 3 weeks through intraperitoneal injections, and then quantified the vascular amyloid accumulation. And what we observed is that in the control mice, the vascular adherence is stained in red and the amyloid stained in green, is that the control mice had robust vascular amyloid accumulation. But in mice treated with cyclosporin CSA or FK506, there was a significant reduction in the vascular amyloid accumulation, suggesting that treating a whole organism with these drugs can reduce cerebral vascular amyloid accumulation. So we were particularly excited about these results because both cyclosporin A and FK506 are used clinically as immunosuppressants post-organ transplant. And there have been some anecdotal studies that have looked at these populations of people and observed that they have lower incidence of dementia than the general population. So for example, here the general population has at age 65 has about a 10% incidence of dementia, whereas people on calcineurin inhibitors have about a 1%. And similar trends continue all the way up to age 85, where the 30% of the general population has dementia, whereas people on these drugs have a 0% chance of dementia or incidence. But these drugs have pretty adverse side effects. They're immunosuppressive, so the people that are taking them are at higher risk for infection and cancer and other side effects. So we wanted to know if we titrated down the dose of these drugs, do we still see improvements in the pathological outcomes in mice? And so we ran this titration experiment where we treated again the mice with a vehicle or 10 mg per kg of FK506, 1 mg per kg or 0.1 mg per kg. And this time, instead of just analyzing the brain for pathology, we also collected peripheral organs such as the spleen and the blood to monitor whether the doses we were applying to the mice was immunosuppressive. And so just to give an example of this, in the spleen, which has a lot of B and T cells localized to it, when we applied 10 mg per kg of FK506, we saw significant suppression of IL-2 and TNF-alpha, which are both immune response genes. And so repression of these genes suggests that the drugs are acting as an immunosuppressant. And we see similar results at 1 mg per kg that IL-2 and TNF-alpha are both repressed. But at 0.1 mg per kilogram, we don't see a significant difference between the control mice that didn't receive FK506. So this suggests that at 0.1 mg per kg, that FK506 is not acting in an immunosuppressive manner. And so this was encouraging, but then we also wanted to know if this led to a reduction in the cerebral vascular amyloid pathology. And so encouragingly, even at 1 mg per kg, we see significant reductions in the amyloid building up along the vasculature. It's not as robust as the higher doses, but still it's significantly reduced. And so this is great that we can reduce the pathology in a mouse brain, but the ultimate goal is to improve cognitive abilities of mice. And so recently we've done some studies to find that even this sub-immunosuppressive dose of FK506 can lead to improved tasks associated with learning and memory. And so we took the 2 cohorts, again, a control treated mouse and a FK506 mouse, and treated them with daily IP injections for 6 weeks. And then we did blinded behavioral studies on these mice led by a talented behavioral postdoc in Liwei Sai's lab, Taeyoon and Kim. And I'll just give you one example of a test that we ran, the novel object recognition test. And basically, if you're not familiar with this test, you place a mouse in a cage where there's 2 objects that are identical, and you allow them to get familiar with these objects for about 10 minutes. Then you place them back in the home cage. And then you put them in the cage where one of the objects has been removed and replaced with a novel object. And the thought here is that mice like to explore. And so if they find the novel object, they'll have a preference for exploring the novel object opposed to the familiar object. And so mice that aren't able to distinguish whether it's novel or familiar have a preference of about 50%. And so the control mice had a preference of about 50%, but the mice treated with the FK506 showed improved ability to recognize the novel object and had a significant difference of about 60% discrimination index. And so we've run a number of tests like this, including the Y maze, the Morris water maze, and we're continuing to do these studies. But consistently, APOE for AD mice treated with low doses of FK506 are showing improvements in tasks associated with learning and memory. And so we're excited to move forward with these results. And so I'll just summarize this project really quickly and say that we were able to reconstruct aspects of the human cerebral vasculature in a dish and then use it to model a pathology associated with Alzheimer's disease. And that allowed us to identify the causal cell, a molecular mechanism, and a way to suppress that molecular mechanism that has now been able to lead to cognitive improvements in animal models. And so most of that story was published in Nature Medicine earlier this year. So if I went through it fairly quickly, please ask questions at the end, or feel free to look at the publication. But next, I'd like to move on to another story that's currently in preparation. And this story really focuses on myelination. And so myelination is this thick lipid layer, cholesterol-rich and lipid layer that wraps around axons. And this is really important for supporting axons and improving electrical conductance. And myelination is conducted by oligodendrocytes, which arise from a long lineage of cells from oligodendrocyte progenitors immature oligodendrocytes, non-myelinating mature oligodendrocytes, and then myelinating mature oligodendrocytes. And a recent study by Bronco recently identified, using single-cell transcriptomics, identified that immature oligodendrocytes and earlier progenitors actually express ApoE at high levels and can be identified in the oligodendrocyte lineage through their high levels of expression. And for a long time, it's also been known that in Alzheimer's disease and dementia, there is reduced myelination. So this is a really old paper, but it's using a stain called Luxol Fast Blue, and it stains for lipid-rich myelinated areas in the brain. You can see in the healthy, there's much darker staining, but in moderate dementia, there's much lighter staining. So suggesting that for some reason, myelin breaks down in the Alzheimer's disease brain. And more recently, single-cell transcriptomic study from Liwei Sai's group, where I did my postdoc, compared the postmortem single cells from the prefrontal cortex, Brodmann area 10, from people that had no AD pathology to people that had AD pathology. And they isolated a bunch of different cells and identified the clusters, which I had mentioned in the intro. But one of the really striking things about this data set is if you zoomed into the oligodendrocyte cluster, the oligodendrocytes from AD patients formed this really unique cluster that was significantly different from oligodendrocytes from healthy individuals. And so this suggests that AD oligodendrocytes, or oligodendrocytes in the AD brain, have a unique transcriptional signature. And so more recently, working with a talented postdoc in Manolis Callas' lab, Jose Davala, grad student Juna, and Leila, we've been looking at single-cell transcriptomics data, comparing people that have the AD risk factor ApoE4 to people that don't have the AD risk factor ApoE4. And this data, we've looked both in the prefrontal cortex and the hippocampus, but I'll show you the data from the hippocampus here, where looking at oligodendrocytes from ApoE4 carriers, we see significant expression differences in genes. What really stuck out here is that a number of myelin-associated genes, PLP1, MAG, MOG, and myelin basic protein, were significantly downregulated in oligodendrocytes from ApoE4 carriers. And if you run all the downregulated genes through a geo analysis, what sticks out is that there's basically two branches of the genes. You have biological pathways and neuronal differentiation and maturation, but the other branch is really focused on myelination and oligodendrocyte differentiation. So this suggests that in human oligodendrocytes, ApoE4 downregulates myelin-associated pathways. And so humans are pretty diverse, or genetically diverse. So we wanted to do a more controlled study. So we looked at the ApoE knock-in mice again, where the mouse ApoE has been knocked out and replaced with either ApoE3 or ApoE4. And so this would allow us to specifically test the role of ApoE4 in myelination. And the classic study for looking at myelination is TEM, or transmission electron microscopy. So we took out the corpus callosum from these mice and compared it, the alter structure of the myelin sheaths in the two different mice strains, or mice with the two different genotypes. And so if you haven't looked at these images before, these blank spots here are basically the barrel of the axon, and it's proceeding perpendicular to the plane of focus. But around the axon are these electron dense areas shown in black. And those are really the myelin leaflets. And what really stuck out to us is that in the ApoE3 mice, there's nice black myelin leaflets that increase with age. But in the ApoE4 mice, the myelin leaflets are more sparse and appear more disorganized. And so the classical way of quantifying this is using something called a G ratio, which takes the axonal diameter to the outer myelin diameter. And so it's a bit of a weird number where 1 equals no myelination, and there have been some theoretical studies that have shown 0.6 equals optimal myelination. And so just quantifying these images, they're qualitatively different. But if you look at the ApoE3 mouse, that has a mean G ratio of about 0.6, which is near optimal myelination. But the ApoE4 mice tend more towards 1, which is lower levels of myelination. And so this really, and some additional human data that I don't have time to show you, really suggested to us that ApoE4 promotes hypomyelination relative to ApoE3. And we wanted to dig into this a little bit more and understand what's going on here. Why does ApoE4 act to reduce myelination? And so to do this, we turn back to our in vitro model systems that allow us to control variables and mix and match genotypes a little bit more clearly. And so here, to build an in vitro model system of myelination, what I did is differentiated the iPS cells into oligodendroglia, or OPC-like cells, and differentiated the same iPS cells into neurons in a different dish, and then mixed them together in a hydrogel, allowed them to interact over a period of time. What we started to see is after about 2 weeks, the oligodendrocytes were expanding through the culture and starting to engage the axon shown in red, and the green stain here is MBP staining. And after about 6 weeks, we see that the axons start to have, the oligodendrocytes start to have, or sorry, the cultures start to have these periodic stains that might be reminiscent of the nodes of Ranvia in vivo. And then we've done some TEM imaging on these cultures, and we see the classical axon barrels with leaflets of membrane wrapping around them. Since we established this model system, we want to go back to our isogenic pairs of ApoE3 and ApoE4, and this time see if we can model differences in myelination in a dish that we'd seen in the mouse model. And encouragingly, what we saw is when we looked at ApoE3 oligodendroglia and ApoE4 neurons, we see a lot of MBP staining that is starting to engage neuronal axons shown in red here. But in the ApoE4 case, we don't see a lot of MBP staining along the axonal tracks. And since we were able to model this in a dish, we wanted to ask, similar to what we did with the mixing and matching in the blood-brain barrier, is if we could pinpoint which cell type was playing a causal role here. And just in the interest of time, I'll show you a simplified schematic where we took ApoE3 oligodendroglia and co-cultured them with ApoE3 neurons, ApoE4 oligodendroglia co-cultured with ApoE3 neurons, where it also included a ApoE knockout co-cultured with ApoE3 neurons. So the only thing here changing is the oligodendroglia cells. What we observed in these cultures is the ApoE3 at around 3 weeks had nice foci of MBP positive areas throughout the culture, but the ApoE4 and the ApoE knockout, we didn't see a lot of these foci developing. And after about 6 weeks, the ApoE3 culture is nicely, a lot of the axons are coated with MBP, whereas in the ApoE4 culture and the knockout culture, things are a little bit more disorganized and the expression of MBP is much lower. We've quantified this at 6 weeks, and we see a significant difference in MBP localized to axons between the ApoE3 and the ApoE4 case. Or significantly, ApoE4 leads to significantly reduced MBP axonal staining. And in this experiment, just to remind you, the only thing that varied was the oligodendroglia. So this suggests that the oligodendroglia have a cell autonomous defect that's preventing them from myelinating properly or MBP co-localizing to the axons. And so to dig into this more again, then we moved to the molecular analysis and RNA sequencing. And this analysis was done by a talented grad student in Liwei Sai's lab, Audrey Effenberger. And she found a number of differentially regulated genes in the RNA-seq data, but a lot of these genes, similar to the stuff that we were seeing in the human brain, were myelin-associated genes such as PLP1, CNPAS, and myelin regulatory factor, so suggesting that in ApoE4 oligodendroglea, these genes are downregulated. But when we looked at the upregulated genes, what really stuck out to us is that cholesterol biosynthesis pathways were nearly uniformly upregulated in ApoE4 oligodendroglea. And so just to zoom into this pathway a little bit more, what I'll show you is this is the cholesterol biosynthesis pathway. And nearly every enzyme in this pathway is significantly more highly expressed in ApoE4 oligodendrocytes or oligodendroglea. And so this was really exciting, suggesting that for whatever reason, ApoE4 oligodendroglea are producing more cholesterol. And around the same time, we made this observation. Jose and Juna were analyzing postmortem brains from ApoE4 carriers. And they came to the same observation that if you look at oligodendrocytes in the human brain from ApoE4 carriers, the cholesterol biosynthesis pathway is significantly upregulated when you have one copy of ApoE4, or even more when you have two copies of ApoE4. These are the same enzymes that we're seeing in our in vitro culture system. So Jose's gone on to analyze this data a little bit more using GeoAnalysis or gene ontology analysis. And it really points to a disruption in cholesterol transport and biosynthesis in ApoE4 oligodendrocytes in the human brain. And so then to start to understand this data a little bit more, we went back to the in vitro models and asked if we could see where the cholesterol is actually localizing. And we thought that perhaps ApoE4 is leading to a mislocalization of cholesterol. So we used different stains called Bo-Dippy cholesterol or Philippin that stain for cholesterol in the cells. And really strikingly, what we observed is that in ApoE4 oligodendroglea in vitro, we see large amounts of cholesterol accumulating intercellularly, perhaps in a lysosome or ER, suggesting that ApoE4 oligodendroglea are unable to transport or efflux cholesterol as efficiently as E3 oligodendroglea. And so there have been drugs such as cyclodextrin, which can help solubilize and dissolve cholesterol and lipid droplets, intercellular cholesterol and lipid droplets. So we reasoned that we might be able to solubilize cholesterol and reduce these cholesterol droplets in the cell using this cyclodextrin, which has previously been reported to. And encouragingly, when we treated the ApoE4 oligodendroglea with cyclodextrin for about a week, we saw that there was a significant reduction in intracellular cholesterol accumulation in ApoE4 oligodendroglea. And so next we wanted to know if you reduce intracellular cholesterol, does this lead to an increased myelination in our in vitro systems? And so we similar to the previous experimental paradigms, we took ApoE3 OPCs with ApoE3 neurons, or ApoE4 OPCs with ApoE3 neurons, but this time treated with a vehicle or cyclodextrin. And in the control-treated ones, we didn't see much myelin foci around 4 weeks, but in the cyclodextrin, we started to see myelin foci starting to emerge around 3 weeks. And then encouragingly, after about 6 weeks, we saw more significant MBP axonal alignment than we did in the control ApoE4 co-cultures. It wasn't a complete rescue back to the ApoE3 state, but it suggests that cholesterol mistransport or impairments in efflux of cholesterol in the ApoE4 oligodendroglia do in part suppress MBP axonal engagement in the ApoE4 case. So I'd just like to summarize this study here, where we've identified that ApoE4 impacts oligodendroglia. And in this case, it behaves similar to a knockout of ApoE, so suggesting that it's a loss of function. And we've been able to identify that ApoE4 impairs cholesterol and lipid transport in oligodendroglia. And this correlates with the reduced myelination phenotype. And just to summarize the previous work I said, presented, ApoE4 in the cerebral vasculature appears to act upon a cell called the pericyte. And in this case, it seems to act a bit differently, where ApoE4 is a gain of a toxic function that leads to increased ApoE around the vasculature and increased vascular amyloid accumulation. And so I think these two stories really highlight the complexities of targeting a single genetic risk factor for Alzheimer's disease. It doesn't seem that it has a single role pathogenic mechanism, but through each different tissue acts quite differently. And so the therapeutic approaches need to take this into account. So I'll just summarize with this slide. My research is really focused on trying to understand why some people live cognitively normal lives into their 80s and 90s, and others, for either genetic or dietary or other lifestyle factors, develop cognitive impairments. And so the model systems, or the approach that I use, is really an intersection between stem cell biology, neuroscience, tissue engineering, and single cell transcriptomics. So I hope I've been able to illustrate these approaches today. So I'd just like to thank all the people that are involved in this work, and in particular, Professor Liwei Sai, who's been a tremendous supportive mentor, Manolis Kellis, who's led the single cell work, and Jose Devala, who's the postdoc in Manolis Kellis's lab, and a bunch of other very talented people that have contributed to these projects and their success. So I just want to thank you for your attention, and if you have any questions, please let me know.
Hello, can I ask a question? Or?
Sure.
Yeah, hi, that's a great talk, really exciting project, congratulations. I wanted to know more specifically about the first part. The endothelial cells basically were shown previously by Zlokovich group to facilitate the clearance of amyloid beta using PCOM clathrin facilitation vesicles. And I don't know if you are careful with your terminology talking about appearance of amyloid beta, but are you touching or thinking about like the clearance in this system, and more specifically, whether in those rescue experiments, whether you try to look at the endothelial cells and to check whether the clearance of amyloid beta that you're seeing so much resembles those pathways?
Yeah, so a lot of that work was very acute. So we applied the amyloid beta and looked, 4 days afterwards. And so I think that it's more, our approach was more designed to look at accumulation versus clearance. The other aspect there, I think, is that it's a static culture system right now. Hopefully we're going to integrate other aspects like the lymphatic system and flow through the endothelial cells. But right now it's static. So I think it's not really geared towards modeling clearance, but I think hopefully in the future we'll be able to do those types of experiments.
Thank you.
Thanks, Joel, for the very, very interesting talk. Any more questions?
Hello, I have one question as well. Joel, it was an amazing talk. Thank you so much for all this information. I'm kind of curious. Have you guys tried to check what is actually inside of the conditional media? So I'm kind of curious, like, what is the main trigger? Like, did you have any like the analysis on that? Thank you.
So for the amyloid accumulation, we ran an ELISA, and it's the conditioned media from AD patients just has a ton more amyloid in it, and so we think that's just the source there. And then we also use pericyte conditioned media, and we've run ELISAs on that and against ApoE, and found that conditioned media from ApoE4 pericytes has much more ApoE itself in it. As far as, you know, an unbiased approach such as mass spectrometry or proteomics, we haven't done anything extensive there, so it could be a more complex answer than we think.
Thank you.
Hi, I have a question. Thank you. It's very interesting talk. You showed the data from the mouse corpus callosum and the ApoE4 mice. I'm wondering if anything similar has been looked for or seen in humans using diffusion imaging. One could, in principle, measure axonal diameters and myelin compaction in the human corpus callosum. Is there anything known about that?
Yeah, I took the slides out because I thought I wouldn't have enough time. But so there's this one study that looked at ApoE3 and 4 in infants. And they found a really interesting observation, is that the ApoE3 infants had much more myelination than the ApoE4. And their conclusion is that some of these risks for Alzheimer's disease might be developmental rather than in age-related neurodegeneration.
Right. That's only in infants, you say, where myelin is probably not mature.
Yes, yeah, I think that's the only samples they looked at. I think there hasn't been a longitudinal study looking at this, I don't think.
Yeah, interesting. Thank you.
Thanks.
If there are no more questions from the audience, I will ask you something. Actually, you have a very impressive correlation, you know, at the molecular level, at the biochemical level, at the cellular and the behavioral level, when you use this immunosuppression. So you do this behavior test in the ApoE4 knocking mice, and then you see an improvement in cognitive tasks, right? My question is, how does this relate to Alzheimer's disease patients? As in, do these mice actually have cognitive deficits similar to the ones that we find in people with dementia? And does the improvement get to a level similar to people who wouldn't be diagnosed with any type of dementia?
Yeah, no, that's a great question. I think the problem with mice is they never develop Alzheimer's disease. So for the mouse models of Alzheimer's disease is pretty extreme. I mentioned it's a 5x FAD, and then we basically have to put 5 things that cause Alzheimer's disease in humans into one mouse. These normally aren't found in one human. And these mice do develop cognitive impairments with, you know, spatial recognition, the novel object recognition, and the Morse water maze. But how that relates to human cognitive impairments is a little unclear. And I think if you look at the clinical trials, the drugs have been able to reverse these things in mice quite effectively, but they've continually failed to do so in humans. So, you know, who knows?
Yeah, well, maybe this is the one. What happens actually if you apply these drugs to perfectly healthy mice? Do they also have cognitive improvement, or is it specific to ApoE4 knocking mice?
I don't think I'm not sure. We haven't done that experiment.
That would actually be a quite good control to have. I would say yes.
Yeah.
All right.
So hello. So can I have a question?
Sure.
Let's go ahead.
Yeah. So is there any effects of ApoE4 on the synapse?
Yeah, there's we don't study it that much, but ApoE4 is associated with hyperactivity and increased LTP. But the sort of the caveat there is a lot of people don't believe ApoE is expressed highly in neurons. So the molecular cause is a little bit unclear.
Can I just ask a brief question about that? Are there any changes in the conduction velocity of those axons measured in any of the models?
Yeah, I don't know. We haven't measured that yet. That's the direction we're going. I think to correlate the structural observations we're seeing with some functional outcomes.
Great. Maybe I'll ask you more later in the session. Does anyone have any more questions before we adjourn for the afternoon?
So I think we can stop here, right? Just a reminder, whoever wants to join, I will have a more extended discussion session at 4 p.m. The same Zoom link will work. So thanks again, everybody, for joining. Thanks a lot, Joel, for the very good talk. It was a pleasure to hear your story.
Thank you for having me. It was a lot of fun.
Okay.
Bye.
Bye, everyone.